Hello, welcome to Census School of Life Sciences. This video is about glycogenolysis. Glycogenolysis is the process of breaking down of glycogen. The major site of glycogenolysis is liver and muscle. All the enzymes for glycogenolysis are present in the cytoplasm. Lysosome also can degrade glycogen through autophagy with the enzyme acid maltase. See, when blood glucose level falls, liver glycogen is broken down and the glucose formed helps to maintain blood glucose level. While muscle glycogen cannot maintain the blood glucose level because the enzyme glucose 6-phosphatase, which convert glucose 6-phosphate to glucose, is not present in the muscle. This will be more clear at the end of this video. See, the key enzyme for glycogenolysis is glycogen phosphorylase, and it is the rate-limiting enzyme. Pyridoxal phosphate is a coenzyme for this enzyme. So in that sense, we can say vitamin B6 pyridoxine is required for glycogenolysis. We can expect such type of questions also in exam. See, in order to study glycogenolysis conveniently, we can divide the process of glycogenolysis into 3 steps. Step 1, action of glycogen phosphorylase to shorten the chain. See, in the first reaction, glycogen is acted upon by the enzyme glycogen phosphorylase. The enzyme cleaves alpha-1,4 glycosidic bonds from the non-reducing ends of glycogen molecule sequentially. It is the non-reducing end. So alpha-1,4 linkage here it is alpha-1,4. This enzyme removes glucose units in the form of glucose 1-phosphate. In the form of glucose 1-phosphate, this reaction requires phosphate, and hence this process is known as phosphorylasis. Important. So glucose is removed in the form of glucose 1-phosphate, and the process is phosphorylasis. So another important thing about glycogen phosphorylasis, it can remove glucose units sequentially until 4 glucose residues remain on either side of the branch point. You see here, let's say 1, 2, 3, 4. Then 2 glucose molecules are removed in the form of glucose 1-phosphate. Here in this branch, 1, 2, 3, 4. Then 3 more. These 3 glucose molecules will be removed in the form of glucose 1-phosphate. Clear? So that means 4 glucose residues on either side of branch point cannot be removed by this enzyme. Then the resulting structure, this is limit dextrin. What you call? Limit dextrin. Then you can see the next step. What happens to this limit dextrin? That is action of debranching enzyme. The debranching enzyme has 2 catalytic activities. 1, alpha-1,4 to alpha-1,4 transferase activity or glucosyl 4,4 transferase activity. Then second, alpha-1,6 glucosidase activity. Simply transferase activity and alpha-1,6 glucosidase activity. The first, debranching enzyme, the transferase activity of debranching enzyme. In this process, the transferase activity of debranching enzyme transfers, as the name implies, transfers 3 glucose molecules, 3 glucose units from one branch to the other. You see, it is transferred to this branch. Then now we have only 1 glucose residue attached in alpha-1,6 linkage. You know, here the branch starting, which branch, which linkage? Alpha-1,6. So this has to be removed with the help of alpha-1,6 glucosidase activity of debranching enzyme. Is it clear? Isn't it? Oh, this debranching enzyme has two activities: transferase activity and the what is the removal of the remaining glucose molecule, that is what we call alpha-1,6 glucosidase activity. Okay. Then next process is the action of glycogen phosphorylase on the linear chain of glycogen. Again, action of glycogen phosphorylase. Here, the debranched linear chain of glycogen. Here, now it become linear chain. Branch is removed. The debranched linear chain of glycogen is again acted upon by glycogen phosphorylase. It removes glucose units in the form of glucose 1-phosphate here also, which is converted to glucose 6-phosphate by phosphoglucomutase. Understand? Glucose 1-phosphate become glucose 6-phosphate. Then what happened this in muscle and liver? In the beginning of the video, I mentioned it. You see, the fate of glucose 6-phosphate in muscle and liver is different. What happens in muscle? What happens in liver? In muscle, it enters into glycolytic pathway. In liver, it is converted into glucose by the action of glucose 6-phosphatase. Okay. Now it is clear. Okay. Thank you.